All right, this is part 2 for your multiple choice section. We're going to talk about genes and alleles. There's only 2 questions that I organized into genes and alleles. The first thing you need to know are anything about alleles. So alleles, genes can have more than one alleles. 2 alleles are on heterozygous chromosomes with respect to that gene, meaning like one can be on one gene or one chromosome and one can be on the other, but they don't have to be identical. They can code for brown eyes or blue eyes, but they're both on the eye color slot on the chromosome. Alleles are found, that's called the corresponding loci, because the location is loci. And there can be alternate forms of the same gene. I already mentioned that. And one allele can be dominant over the recessive allele. And then you have one more question that I wrote down for this one. And it says, give an example of alleles. So an allele would be like a big T and a little t, or a big A, or a little a, or a big B, or a little b. Those are alleles. And then the next one we have is monohybrid crosses. So we have a few questions about that. If I give you a cross between 2 mice and both of them have, let's say, blue fur, we're going to make blue fur mice. And we'll say blue fur is dominant over yellow fur. And 75% of the offspring end up having blue fur, and 25% of them have yellow fur. What can you assume the parents' genotypes are? So you would make the Punnett square, and you would say that, 75% of them had to be blue. So you had to have a blue here, and a blue here. Out of 4 boxes, that's 3 of them would be 75%, and 1 of them would be yellow, so it's going to be little b's. So what's the way that we do that? Well, we know there has to be a little b here, and we know there has to be a little b here in both of those spots, right? That's how that works. Because one parent had to give a little b and the other parent had to give a little b. Give a little b, give a little b. But then this one has to be a big B because of this and this. And then this one had to be also a big B. So each parent would be big B, little b, or heterozygous. And the next one I had for monohybrid crosses was this question. Again, if you are recessive for a trait and you show up the recessive phenotype, then you have to have 2 lowercase letters in order for that recessive phenotype to show up. That's a pretty straightforward one. And this one, the words heterozygous and homozygous. So, what was the number again? I'm looking. So a genotype, you could have a heterozygous genotype, which looks like this. You could have a homozygous genotype that looks like this, or like this. Those are all heterozygous or homozygous genotypes. But you have to have the letters, and you have to have 2 letters in order to be a genotype. A single letter is considered an allele. You could also have multiple letters. You could have letters like that. And this is still a genotype, it's just a genotype for 2 different traits. Now we're going to talk about meiosis. Remember, meiosis is the process of one cell going through 2 divisions to make genetically different daughter cells that end up being haploid or having half the number of chromosomes that you do to begin with. One of the unique things that happens during meiosis is the concept of prophase, crossing over in prophase 1, So crossing over is when genes from one chromosome will end up on another chromosome. So all of the DNA from mom's chromosome doesn't always stay on mom's chromosome. Sometimes it ends up on dad's chromosome. And that's an important mixture of genes that you could potentially get. It greatly increases the number of varieties of offspring that you get. I have to scroll way down because these were at the bottom of his questions. Again, they're going to be all mixed up for you, so don't get all excited. The difference between meiosis 1 and meiosis 2, one of the differences is that meiosis 1, when it first divides, it's splitting homologous chromosomes, and then sister chromatids, which are like connected at the centromere, will split during meiosis 2. And so that means meiosis 2 is already haploid when it splits again. Meiosis 1 does not produce diploid cells. Meiosis 1 produces haploid cells. They line along the equatorial plate in all of them. And so there you go.
Mom, can I have ice cream?
Types of reproduction, evidenced by diploid or haploid forms of an organism. Basically, if you want to know how the reproductive cycle of an organism works, you look to see at which stages they are diploid and at which stages they are haploid. That seems kind of weird for a human to talk about because humans are only haploid when they're a sperm or an egg. They're not even a full organism at that point. But other organisms spend a long part of their life as haploid organisms. I think funguses, for example, are some of those, or sporos, some organisms that reproduce with spores, like fungi might do that sometimes, where they spend part of their life as haploid organisms. So I'm going to stop this video here, and then we'll do probably 2 more. I'm trying to keep them short.